present to you our results from our multi-center randomized phase 2 trial of granulocyte macrophage colony stimulating factor, GM-CSF, plus ipilimumab, maybe referred to as IPI, versus IPI alone in patients with metastatic melanoma. This is the Eastern Cooperative Oncology Group trial, ECOG 1608. So cytotoxic T lymphocyte antigen 4, or CTLA-4, is a natural break to the immune system. And the fully human monoclonal antibody, ipilimumab, is an antibody that blocks CTLA-4 function and that essentially takes the brakes off the immune system. Ipilimumab improves survival in patients with metastatic melanoma and is currently FDA approved for the treatment of patients with metastatic melanoma. The question then becomes, how do we improve on these results? GM-CSF is a chemical signal in the immune system that enhances granulocytes and macrophages. It is currently approved for bone marrow support following high-dose chemotherapy. In preclinical animal models using mice, CTLA-4 blockade plus a vaccine, a whole cell vaccine, that secretes GM-CSF had demonstrated synergy, where the combination of treatment had better efficacy than either treatment alone. So this is the schema for ECOG 1608, where 245 patients were randomized with either unresectable stage 3 disease or stage 4 disease. And they had to have either not received treatment for metastatic disease or up to one prior therapy. In Arm A, 123 patients were randomized to receive sargramostim, which is also GM-CSF, at a straight dose of 250 micrograms administered subcutaneously daily on days 1 to 14 of a 21-day cycle. They also received ipilimumab at 10 milligrams per kilogram intravenously on day 1 of each cycle times 4 doses, and then every 3 months at the same dose, a so-called induction and maintenance regimen. This regimen is different than the currently FDA-approved regimen of 3 milligrams per kilogram intravenously every 3 weeks times 4 doses. 122 patients were randomized to Arm B, which essentially is the ipilimumab induction and maintenance schedule as an Arm A, but treatment with IPI alone. The addition of sargramostim to ipilimumab improves overall survival with a median follow-up of 13.3 months. Here you can see the Kaplan-Meier curves with IPI plus GM-CSF in red and IPI alone in blue. The stratified one-sided log rank p-value of 0.014 offers a hazard ratio of 0.64, a 36% reduction in the risk of death. The one-year IPI overall survival rate was 52.9% versus 68.9% in the combination, and the median overall survival for IPI was 12.7 months versus 17.5 months in the combination. Also, the addition of sargramostim to ipilimumab offered improved tolerability with a less incidence of serious side effects with the addition of GM-CSF to ipilimumab. Specifically, there were less gastrointestinal and pulmonary serious side effects that were witnessed. So in conclusion, adding sargramostim to ipilimumab improves survival and tolerability. With both drugs commercially available, there are implications for current treatment for melanoma patients as the addition of GM-CSF improved efficacy as well as side effect profile. However, of note that the dose of IPI used in this trial was 10 milligrams per kilogram, included an induction and maintenance phase. Currently, there's a trial that has been completed to compare 3 milligrams per kilogram to 10 milligram per kilogram with ipilimumab that has completed accrual, and we're waiting for the data to become mature. Importantly, though, I think these data represent an important avenue for combining cytokine therapy with immune checkpoint blockade and the consideration of the future use of GM-CSF and other immunotherapy development, such as with combination of PD-1 blockade. Thank you very much for your attention.